Hello and welcome to Nikolay's genetics lessons, and today's problem is you have performed a dihybrid test cross for two genes A and B. The following offspring were produced, and here is a ratio and genotypes. Question A: are the genes of the dihybrid in coupling or repulsion? And question B: what is the percent of recombination between these two genes? First of all, let's check the ratio that we have here: 10 to 40 to 40 to 10. If gene A and B would be on the different chromosomes, then we are going to get 1 to 1 to 1 to 1 ratio, or 25%, 25%, 25, and 25%. Because we see that some genotypes we see more frequent than the other genotypes, we can say that gene A and B on the same chromosome. And the closer gene A and B to each other, then the number of recombinants would be smaller. So this genotype and this genotype stand for the recombinants. If, for example, two genes would be, gene A and B would be on the very tip of the same chromosome, then number of crossing over between these two genes and number of recombinants also would be the same as if these two genes would be on the different chromosomes and would segregate independently. So again, we would see numbers which would resemble independent assortment. So we would see 25%, 25, 25, and 25%. Now, when we know that gene A and B are on the same chromosome, still two variants are possible. Take a look. The first variant would be here's one chromosome, here's another homologous chromosome. And here we have dominant gene or allele A and dominant allele B. And on the other homologous chromosome, let's say we have recessive allele A and recessive allele B. And we cross, because this is test cross. Take a look. Let's say this is individual, person, animal, or plant. For the gene A, this individual is heterozygous, capital A, small a genotype. And for the gene B, this individual also heterozygous, capital B, small b. Dihybrid means that for gene A, this individual is hybrid, and for the gene B, also hybrid, so dihybrid. But also we are told that it is a test cross. And test cross would be if second individual also would have the same chromosome. Let's say it can be chromosome number 1 or chromosome number 2, but second individual for the same chromosome also would be following genotype: small a, small b here, small a, and small b here. Now this cross we call test cross. In a test cross, one would be heterozygous for the gene A and B, and another individual would be homozygous recessive for the gene A and B. Let's also put parent 1 here and parent 2 here. And this parent can produce gametes, and this individual also can produce gametes. In the gametes, of course, we only can find one chromosome from this pair, and individual 1 also would give only one chromosome out of this pair. Now imagine that between gene A and B, crossing over may happen or may not happen. Only two variants are possible. Crossing over happened and crossing over didn't happen. So let's find in the F1 progeny, in the following progeny, what variants are possible if crossing over didn't happen. For example, parent 1 would give this chromosome and parent 2 is going to give this chromosome. So in the F2 generation, we are going to get following genotypes. So capital A and capital B chromosome inherited from the parent 1, and small a and small b, this is going to be chromosome inherited from the parent 2. We also may get following variant, for example, this chromosome and this chromosome. But again, genotype is going to be the same, because the same chromosome inherited from the parent 1 and second chromosome, which is homologous to the first one, still have the same genotype. So these two variants would produce the same genotype. We also can get another variant, for example, this chromosome from the parent 1 and this chromosome from the parent 2. Again, this chromosome from the parent 1 and this chromosome from the parent 2 will produce the same genotype, which is going to be as follows. So small a and small b from the parent 1 and small a and small b from the parent 2. Again, this is genotypes, this is variants, which we can get in the F1 generation if crossing over between gene A and B didn't happen. In the next generation, we will find one individual heterozygous for the gene A and B, and other type of individuals we can find which are going to be homozygous recessive for the gene A and B, and they would occur with the same frequency, 50% this genotype and 50% this genotype in the following F1 generation. But now let's assume that crossing over may happen between gene A and B, which is actually happens all the time, and let's say we have a crossing over here. Then parent 1 is going to produce following variants. For example, recombinant chromosome from the parent 1 would be capital A and small b. So let's put it here. So capital A and small b. And from the parent 2, only one variant of the chromosome possible. Even if crossing over happens here, still it's not going to change anything. Let's say crossing over happened over here. So first variant parental was small a, small b. Now we have crossing over which happened here. We still have small a and small b, small a and small b. As you see, crossing over in this parent who is homozygous recessive for the both genes doesn't change anything in our calculations. So parent 2 only can give following variant of the chromosome with recessive allele A and recessive allele B. And when we have crossing over here, it creates new chromosome with capital A and small b, but also creates another chromosome, recombinant chromosome small a and capital B. Let's also add this variant here. So this is going to be small a and capital B, and parent 2 only can give one variant, which is small a and small b. The first two variants we say are parental variants, because in every combination here we see one chromosome from one parent, another chromosome from the other parent, one chromosome from the parent 1 and another chromosome from the parent 2. So these variants always would prevail in a progeny. And these variants here, which I circle with red color, would be variants which were produced by recombination. And of course, such events would be rarer, because only if two genes would be at the very tip of each chromosome, then we expect that recombination would be so frequent that we cannot tell the difference if, for example, these two genes would be on the different chromosomes. But the closer two genes which are on the same chromosome, then the least probability that crossing over is going to happen between them, and that means that number of the recombinants will be always smaller than number of non-recombinants. At this point, you would be able to tell whether gene A and B are coupling, meaning both dominant alleles are on the same chromosome, or in repulsion, meaning that on one chromosome dominant allele A and on the other chromosome dominant allele B. So here we have dominant A and recessive B, and here we are going to have small a and dominant B. So this situation we will call repulsion. Sometimes we can use different terminology, cis and trans, just like in chemistry. But for your deep understanding, I want to continue and show you the same picture which we are going to get if parent 1 would be heterozygous for the gene A and B, but these genes these alleles would be in repulsion. And then I will show you a shortcut how to solve this type of problems in just seconds. But in order to do so, you have deeply understand this type of problems on the chromosomal level and have idea how crossing over affects our calculations. So let's say we have different situation. So parent 1 have two homologous chromosomes. So parent 1, but this time we assume that dominant allele A is going to be here and recessive allele B here. Recessive allele A here and dominant allele B here. And this is going to be parent 1, and parent 2, as you remember, is just going to be homozygous recessive for both genes. So it's going to have small a, small b here, small a, small b here, and this is what we call dihybrid test cross. Now, as you see, this is what we call repulsion or trans situation with two genes, one being dominant, another recessive, on the same chromosome. Still, parent 1 would be heterozygous for the gene A and heterozygous for the gene B, just like in our previous example, but this time it's going to be in repulsion or in trans configuration. Now let's imagine that no recombination is going to happen. First variant would be that parent 1 would give this chromosome and parent 2 is going to give this chromosome. Again, we can also combine these chromosomes and we are going to get the same picture, the same genotype. So chromosome inherited from the parent 1 have genotype capital A and small b, and from the parent 2 small a and small b. And the second variant, which is possible, that this chromosome come from the parent 1 and this chromosome come from the parent 2. Or again, this can be the same chromosome from the parent 1 and this chromosome from the parent 2, but as you see, it doesn't change anything. So we are going to get following variants in the F1 progeny. So we are going to get small a and small b here, small a and small b here. One chromosome came from the parent 1, another chromosome, it can be this one or this, came from the parent 2. This variance we can get if no recombination happen between gene A and B. And we call these chromosomes parental chromosomes. But also again, what might happen that between gene A and B, crossing over happened because they are on the same chromosome. So crossing over may happen. In this case, parent 1 would produce new variant, new recombinant chromosome, where we'll find dominant allele A and dominant allele B on the same chromosome. And this is going to be recombinant chromosome, capital A and capital B. And parent 2 only can give only one variant of the chromosome with recessive allele A and B on it. And if we got such crossing over, that means that we also can get another recombinant chromosome with recessive allele A and recessive allele B coming from the parent 1. So here is going to be another variant, and this is going to be recessive allele A and recessive allele B. And from the parent 2, we only can get one variant, recessive allele A and B. And as you see, this is going to be a result of recombination. So these genotypes would prevail in a progeny, and this result of recombination we will find in a fewer numbers. Now let's write down genotypes. For example, here's genotype of the progeny for the gene A, which is capital A and small a, and the same individual would going to be also heterozygous for the gene B, capital B, small b. We are going to find this genotype in most of the cases. Also, we are going to find a lot of this genotype, which is going to be small a, and small b, small b. So small a, and small b, small b. So we will find this in larger numbers than, for example, these genotypes, which are going to be capital A, capital A and small b, small b, small b, small b. And this variant also small a, and capital B, small b. If parent 1 would be dihybrid for the gene A and B, but dominant genes would be on the different chromosomes and recessive genes would be on the different chromosomes, or we also say alleles. In this case, what prevailing genotypes in a progeny going to be? We know these genotypes. Now let's write them down. One gene A would be heterozygous, so capital A and small a, and another would be homozygous recessive in the same individual, so small b. And we are going to see this genotype in most of the cases. Another variant would be small a, and small b. So small a, and small b. We are going to see also a lot of this genotype. And we are going to see these genotypes in fewer cases in a progeny. Capital A, small a, and capital B, small b. So let's write down capital A and small a, and capital B, small b. And another variant would be this genotype, which is small a, and small b. Now let's check the frequency of the genotypes, which we were given in our problem. And we see a lot of this genotype and this genotype in a progeny, which stand for the non-recombinance. And take a look. This genotype is the same as here, and this genotype is the same as here. So this in much fewer quantity, and this genotype is also in much fewer quantity. This tells us that parent 1 have this genotype. It is in repulsion state. So it is heterozygous for the gene A and B, but dominant allele A and B are on the different chromosome, not on the same. So we also can say in trans combination or in transform. So let's check our problem again. The answer to the first question would be that parent 1 is dihybrid and genes are in repulsion. So let's circle it. This is going to be our answer. And as for the second question, what is the percent recombination between these two genes? As I said earlier, if gene A and B would be at the very tip of the chromosome, maximum number of recombination would be 50%, whether recombination happen or not. Whether it happens, for example, twice, then in this case we are not going to get a recombination. If it happens once, we are going to get recombination. If it happens 3 times, we are going to get recombination. So even number here means no recombination. Uneven number means recombination if crossing over happened between gene A and B. So you should understand that number of recombinants cannot exceed 50%. One more time, if the gene would be at the very tip of the chromosome, they would behave as if they are on the different chromosomes. And we are going to get ratio, for example, 25% for this genotype, 25% for this genotype, 25% for this genotype, and 25% for this genotype. All genotypes would be represented in the following generation in about equal quantity with a ratio of 1 to 1 to 1 and to 1. As you see, 50% maximum number of recombinations between two genes on the tip of the chromosomes means that one chromosome cannot be greater than 50 centimorgans. One chromosome cannot be, say, 100 centimorgans. Maximum it can be 50 centimorgans. Those all chromosomes, for example, in human genome, chromosome number 1, 2, 3, 4, 5, 6, 7, they all different lengths, but when we say in centimorgans, their length is going to be the same, 50 centimorgans, because this is relative measurements, which is true for each chromosome. But it's going to be 1 centimorgan for each chromosome would be different measure in, say, Armstrongs. Because numbers today are convenient, together they would add up being 100. It is easy to say that number of this recombinants plus number of this recombinants, which is 10, together is going to be 20. So 10 plus 10 of each recombinant kind would give us 20%, which also equals to 20 centimorgans. This is distance between gene A and B. So distance is 20 centimorgans out of maximum 50 centimorgans if genes would be on the tip of the chromosome. So this gives us information that probably this genes position at one gene here and another gene here. So this is relative position of the gene A and B to each other, relative to each other. And now shortcut method, as I said, you can solve this problem in seconds. So these two highest numbers represent parental genotypes. And genotype is going to be as follows: capital A and small b for one chromosome, and another one comes from the test cross from the second parent, small a, small b, you know it. And another one would be small a and capital B. Again, another chromosome comes from the test cross from the second parent who is homozygous recessive for the gene A and P. So now we know that this genotype represents parent 1, and we know that it is in repulsion. And this is all for today. Subscribe and see you in the next video. Goodbye.